cuando empiece.
Okay, welcome to this week's Museo Nacional de Ciencias Naturales research seminar. So this week we are very pleased to have with us Frédéric Fyon, who works on the role of natural selection in the evolution of processes associated with reproduction. So Frédéric did his degree and his master's in life sciences and in ecology, biodiversity and evolution at the École Normale Supérieure in Paris. He then went to the Center for Functional Evolutionary Ecology in Montpellier, where he did his PhD, and he graduated from his PhD in 2016. Frédéric then spent 3.5 years volunteering in France and Mexico in projects relating to social inclusion, sustainable development, and humanitarian relief. Great, well done, Frédéric. And then decided to return to science, where in 2020 he began a postdoctoral research position at Royal Holloway University of London, working on the evolution of asexuality in a freshwater fish called the Amazon molly. And I think that's going to form part of your seminar today. So thank you very much, Frédéric, and I'll give you the floor now.
Okay, there we go. All right. Well, hi, everybody. Thanks for being here to listen to this talk. I wanted to take a couple minutes before starting with science to introduce myself a little bit. So I'm Frédéric. I live in this beautiful city of Cuenca, just 1.5 hour drive from here. And, well, let's say I'm looking for job opportunities and collaborations in the area, so that's why I came to visit the museum today. A bit about my profile. I'm an evolutionary biologist. I'm a theoretician. So I build mathematical and numerical models to understand evolutionary patterns, or to basically to know how and why things are the way they are today, right? More precisely about my interests, let's see. No, this is not working at all. And if I'm doing here, yeah, okay, let's do that. Well, I'm going to do it this way, no problem. So about my interest more particularly. I'm interested in everything that has to do with sex in a broad sense and selection. So my research is broadly organized into 2 axes. First, I look at features of sex and how they evolve or are influenced by natural selection, among other factors. So for example, I've worked on sex chromosomes, and we developed during my PhD a new theory to understand why sex chromosomes evolve the way they do. And more recently, I've worked on recombination hotspots, trying to understand their evolutionary dynamic and also trying to understand their evolutionary origin. Why do we have recombination hotspots? And the second axis of my research has to do more with alternatives to sex, and again, how they evolve or are influenced by natural selection. So, for example, I've built a model in outcrossing and self-fertilizing species, looking at how mutation load builds up differently in these two kinds of species, and more importantly, how these loads, these different loads, would influence the genetic ancestry of the hybrids. And also with Francisco in London, I worked on asexuality and hybrids. And that's really what I'm going to present to you today. So why would hybrids turn down sex? Here, right now, I'm going to try to convince you that we are developing a new theory to understand the evolutionary link between hybridization and asexuality. And I'm going to do something very cliche. I'm going to start with a quote of Darwin, of course. I just put up this quote to underline that since the very beginning of evolutionary biology, we wouldn't understand why sex. Why we would bother doing sex. Because it's so costly, right? You need to find a mate. You have it's dangerous, whatever. When it's so much easier to just do parthenogenesis and produce an individual, a progeny, right on, right? So this was seen as a central paradox of evolutionary biology for decades and decades, and a lot of research has been done on that, right? And yes, and I think right now we are at a point where we do understand why sex. And I just wanted to summarize this very quickly. You have basically two broad explanations of why sex. You have first, I think now we know why sex is beneficial, and that's mostly due to the great work of Saliotto. Just a quick explanation about that. You need to accept right now that genomes in nature, they tend to be suboptimal, right? They tend to be a mixture of good and bad genes, and that can be due to many different processes such as random drift, selective interference, red queen dynamics, local adaptation and migration, you name it. So you have this mixture, and what sex does is shake it all up, and you have, it recreates the best haplotypes and also the worst haplotypes. But what happens here is that the worst haplotypes are going to get eliminated very quickly because they are so bad. So the genes of sex that are going to endure will be the ones that will be associated with these great haplotypes here. So you can see straightforwardly that genes of sex are going to be associated statistically to better genetic background, and they're going to win, they're going to gain a fitness advantage from that. And that's the main advantage of sex on the short term, right? And so, okay, so then if you accept that sex is broadly great, it has evolved at some point ancestrally in eukaryotes. But the question is, maybe sometimes asexuality is better. It can happen still, right? So there is another kind of explanation that explains that it should be actually hard to switch back to asexuality, even when asexuality is better, right? That's easy to understand because sexual species, they have been sexual for a very long time. They have a huge spectrum of adaptations to sex, right? So this means that asexuality, when it arises, is going to face a lot of physiological constraint. I guess the best example of that is the egg activation constraint, which is something very widespread. So when you have a sexual female that produces some eggs, right? These eggs cannot enter embryogenesis on their own. And that's to prevent that haploid eggs enter embryogenesis and produce haploid offspring. Generally, what you need is the entrance and the fusion with the sperm to activate the egg, and at this point, it will enter embryogenesis. But then you can see that if you have an asexual female that still retains this constraint, there is no sperm, there is no male. So, okay, the female produces a great diploid egg, okay, but it cannot enter embryogenesis. It cannot give birth to a new individual because of this constraint. And actually, there's like a list of other constraints like that, where, yeah, asexual species are going to face big obstacles if they want to spread. So overall, what this picture gives us is that asexuality should not evolve easily, right? And we can now understand why sex is so widespread in eukaryotes. And I think we even are at a point where the paradox has in some way reversed. Because when you look at the eukaryote tree, you actually have a lot of asexual species. Well, not a lot, because it's still a small percentage, right, of all eukaryotes. But the point is that here I've put in colors all the clades where you can find some asexual species. And it's basically everywhere. Asexuality has evolved multiple times in eukaryotes. Right? You can find it in zooplankton, you can find it in insects, you can find it in worms, and even in vertebrates. Right? And even at the small scale, for example, if you look at this little stick insect tree here, Timema, in Timema you have at least 5 different evolution of asexuality. They are independent evolution. So if you sum up all the eukaryotes, you have really asexuality that has evolved many times. And that's the paradox, right? I just explained that asexuality should not evolve easily in eukaryotes, and still it has evolved repeated times. So how can we understand now this paradox? How can we explain that asexuality comes again and again and again? It's probably an answer that has many factors, many answers actually. And probably one key has to do with hybridization. So if I take again this little tree here, I've put in this circle, so the numbers are not from me, like the percentage of the asexual species that are of hybrid origin. Okay. It's a small number of species, so it may not be entirely representative, but still, it gives a good idea, especially in vertebrates. If you look at vertebrates, what we know of vertebrates, all vertebrate species, asexual vertebrate species we know of are hybrids, except maybe one lizard that it seems that it's not hybrid. It's the only case we know where it's not a hybrid. So really, hybridization seems to really be a key factor in the emergence of asexuality. And obviously, what we want to know, what we want to understand is why. What is the link between asexuality and hybridization and asexuality? So there is already a theory that is not evolutionary. We have a mechanical theory where, well, people are saying, okay, well, you have 2 sexual species, A and B, and they hybridize. And often when you have hybridization, the hybrid is weird. So it has some problems. And just by mere chance, sometimes the problems turn out to be some meiotic disruption, and you end up producing a diploid egg because by chance. And sometimes you're going to have problems in fusing the egg and the sperm, so the sperm is not going to fuse with the egg, and the diploid egg is going to produce a clone, right? And you end up with a straightforwardly asexual F1 individual. So here the whole idea is just, it's mechanical. Sometimes genomic incompatibilities in the parental species are going to produce a straightforwardly asexual hybrid. And that's it, right? It seems a little bit far-fetched, and yet it has been seen in nature and in the lab, right? So the classical case is this European spine loach in the Eastern Europe, where we have seen in the wild and in the lab, indeed, these F1 females that will produce diploid non-recombinant eggs and give birth to clones straightforwardly. So we know it works, right? But it may not work all the time, and that's what now I'm going to try to convince you about. So what we did was take the example of this little fish of the Gulf of Mexico, the Amazon molly. The Amazon molly is Actually, it is the first ever reported unisexual vertebrate in early 20th century. And it's a hybrid that has but it's a hybrid between these two species of molly, the Celphine and the Atlantic molly. And it has been around, it's estimated at least 100,000 years. So it's been around for a while. And like many asexual species, the Amazon molly does a gynogenesis, or sperm-dependent parthenogenesis, which means that the way it found, let's say, to circumvent, for example, the egg activation problem, right, is that you have a diploid egg, right, produced by the Amazon female, and it needs a sperm to activate and enter embryogenesis. So what it does, it mates with males from the parental species. It receives a sperm, right? The sperm actually fertilizes the egg and enters the egg. But at this point, you have a process that is not entirely known. We don't really know physiologically what's happening there. But this paternal genome is eliminated, right? And in the end, you get a diploid clonal, perfectly clonal offspring. People have been, we take the example of the Amazon molly because it's a well-known fish. It has been bred in the lab for a long time. And people have been trying to recreate the Amazon molly many times. So what they would do, they would just like cross the parental species, so the Celphine and the Atlantic molly. You cross them, you get a lot of offspring, and interestingly, this offspring is female biased for some reason. And they would take these F1 females, and they would have them mate with black mollies. And the point of mating them with black mollies is that black mollies tend to have black progeny. So if you have black pigmentation in the descendant, then you can say, well, it seems like the paternal, in that case, the paternal genome, the genome of this fish, has been transmitted to the progeny, right? And they would obtain 100% of black-spotted individuals, right? Interestingly, you have only 1/3 of these individuals that are normal diploids, and 2/3 that are triploids. So it seems that these females already have some capacity to produce diploid eggs. But still, you cannot say that they are asexual because these are definitely not clones, and the genome of the father has been transmitted, right? So they are definitely not readily asexual F1s. And this has been done many times, and never ever have we been able to get readily asexual F1 in this case, in this species, right? So, of course, if in some cases, so there are also some other data in other species, in stick insects, in Drosophila, where you have maybe some evidence of back-crossing. So obviously, if you have evidence of back-crossing in the hybrid lineage, you cannot say it was straightforwardly asexual from the beginning, right? So if in some cases at least, and we don't know how many, but in some cases, the mechanical current theory seems to not work. So then if you have an F1 that is not straightforwardly asexual, you need an evolutionary link, a path to explaining how then you're going to end up having a completely asexual hybrid lineage. And so that's what we were interested about, basically. And to answer that, to find that route, what we decided to do was to do some adaptive dynamics. So now I'm going to guide you through the model we made and why we made it this way. So when you do adaptive dynamics, first you want to know what you're talking about. You want a demographic setup. Okay? So we model the two parental species and the hybrid species, the populations. And we're going to assume that females from the different species or different populations have a certain preference. So for example, females from these species have a preference CP for males from the same species. Okay? And then in that case, you just give birth to an individual from the same species. Or it also can mate and prefer a male from the other species, the other parental species. And in that case, obviously, you're going to produce a hybrid, right? And the hybrid females also have a preference. So hybrid females prefer with CH hybrid males, or they can backcross with the parental species and produce also more hybrids. Okay. And with these parameters CH And once you know the relative amounts of males from the different population, then you can easily calculate mating probabilities. So I have a parameter here of phi ij, which is simply the probability that a female from population I is going to mate with a male from population J. And that's a parameter that then later is going to be very useful to understand in which conditions I'm going to have asexuality arising. Okay, so that's your demographic setup. And obviously you need to know, we're talking about evolution. So what are the phenotypic traits that are going to evolve? We want to know about asexuality. So we put 3 traits. First, the capacity to produce diploid eggs by clonal eggs by apomixis, let's say. That's alpha. Then you have the capacity to eliminate the paternal genome, which is the other important trait of gynogenesis, right? So that's the trait beta. And finally, we're going to assume that sex ratios of the progeny is also a phenotypic trait that is coded genetically. So you have a third trait, sigma, which is the sex ratio of the offspring. A fully asexual individual will have alpha equal 1, beta equal 1, sigma equal 1, right? It will always produce clonal eggs, always discard the paternal genome, and produce only females. Right? And now we get to the core of the model. We made a very important assumption right now. I'm going to explain you why. This is just one of the models we made, okay? We made other assumptions with other models, and in the conclusion, I will talk to you a bit about that. But for now, we're going to look at this model because I think it's nicely illustrative. What we're going to assume is that, so we're talking about a hybrid, right? Hybrids are weird again, they have some problems. And here we're going to assume that the problem of the hybrid males is that their sperm is not entirely functional. So the sperm exists and can move around and can fertilize, but let's say it has some problems with the decondensation machinery, for example, and it cannot decondense and fuse with the genome from the egg, right? So what are the consequences of that? If you have a meiotic egg that is going to mate with a hybrid male, receive hybrid sperm, this sperm is not, in the end, it will be destroyed because it cannot fuse with the genome of the female, right? So you will end up producing a haploid offspring, and we're going to assume that this is unviable. This is going to die. So a meiotic egg, to produce a viable offspring, will have to receive a parental sperm, which is a normal sperm. The genome can normally decondense and fuse with the egg. That's no problem there. So you produce a diploid offspring. Except if you have evolved paternal genome elimination. In that case, you eliminate actively this progenome of the sperm, and again, you produce a haploid offspring, and that's bad. Okay? And obviously, if you have an apomictic egg, so a diploid egg, right, the contrary happens. If you receive a hybrid sperm, this goes nowhere, and you still have a nicely diploid offspring, and everything is fine. But if you receive a parental sperm, you receive the progenome of the sperm, then you produce, oh, I forgot a little, there should be 2 there. Anyway, you produce a triploid offspring, and again, we're going to assume that this is bad, right? So if you are in this branch, if you have an apomictic egg that receives a parental sperm, then you're going to need to put paternal genome elimination into action to still produce a nicely diploid offspring, okay? That's very important, and we're going to see soon why. Anyway, so now we have basically our basis that is laid out. We need a question, of course. We want to know, remember, why asexuality is going to evolve. And we're doing adaptive dynamics. So this means, in terms of adaptive dynamics, that I want to know what are the conditions for an asexual mutant to invade, right? If I have very few mutants, what is the condition for this mutant to invade the population? So to do that, we have our demographic setup, and we're going to divide the hybrid population into 2 kinds of individuals. I have my Y-type normal hybrids, and I'm going to assume that these normal hybrids are completely sexual. Okay? So they have alpha, beta, sigma equals 0, right? They don't produce unreduced eggs. They don't do paternal genome elimination. They produce unbiased sex ratios. And then I can have mutations, and I can produce mutant hybrids. And these mutant hybrids are partially asexual, which means they have a certain phenotype where these are not all 3 zeros. Okay? And so what I want to know in terms of mathematics is when do I have the derivative of the population size of these with respect to time that is greater than 0, which means when do I have the number of mutants that increase? That's it. When I have very few mutants at first, right? Are they going to go extinct, disappear, or are they going to invade? I need to calculate a few things before reaching that. First, I need to know how many parental individuals I have around. I'm going to assume that the parental populations are very widespread and large, so whatever happens in terms of population dynamics at the hybrid level is not really going to impact what's going on in the parental species. Like parental species are these big pools of individuals that don't move, that are not influenced by hybrids. That allows you to calculate something that is very classical in population dynamics, whatever. We have our parental population size at a constant number, right? And then I want to know how many normal wild-type hybrids I have around. So right now, I'm not talking about mutations yet, right? So I have no mutant, and knowing how many parentals I have around, I'm going to calculate how many hybrids I have around. So again, you have a classic population dynamics equations. I just put that here to show you how these hybrids, these wild-type hybrids, are produced by direct hybridization between the parental species, or they are produced by backcrossing, and they are removed by death. And that's it. That's very simple. So to find the equilibrium, you just put this derivative to 0, and you find your number here. And you calculate it, and you're happy. So what you have in terms of population dynamics, only you had the parentals at first, right? And then for some reason, they started to hybridize. And so your number of hybrids has grown and reaches an equilibrium, right? And now we are there. We are at this equilibrium. And I'm going to put some mutants, right? So I'm going to do the invasion analysis. So I put some mutants, very few. In terms of mathematics, that's going to mean that the number is going to tend to 0. And I'm going to look at if the number increases, if the derivative is greater than 0. So when I do that, I obtain this very beautiful equation that we don't really care about. I just put it there because with that, I'm going to prove something very interesting. Here I have something that is greater than 0 times something that is greater than 0 times alpha 2, strictly greater than 0. And what I just proved with that, without caring at all about what this is all about, is that I need alpha 2 strictly greater than 0 for the number of mutants to increase. This means that the mutant must have a non-zero rate of apomixis. It must produce some unreduced diploid eggs, at least some, right? And the mutant is going to invade if and only if it produces some unreduced diploid eggs. So if you imagine that a mutation, one single mutation, can only modify one phenotypic trait at a time, I just proved that it needs to modify alpha, right? So I'm going to just put beta and sigma equals 0. I can simplify a lot the equations, and I can prove that another very important condition for the invasion of the mutant is phi HH equal 1/3. So what is that? This is the probability of a hybrid-hybrid mating. Okay? So I need a certain rate of hybrid-hybrid mating for the apomictic mutant, partially apomictic mutant, to invade. Okay? Why do I need these hybrid-hybrid matings? Let's go back to this little figure. So I'm talking, what is the benefit of producing some apomictic eggs? It must have one because the mutant is invading. You have this apomictic egg, you can see that, so you don't have paternal genome elimination yet, right? Remember, we put that to 0, so you can cross that. So obviously, you can see straightforwardly that the only way for an apomictic egg to produce a nice diploid offspring is to mate to receive a hybrid sperm, right? Because the sperm will not be incorporated, the sperm's genome, and you will have still a diploid offspring. If you receive a parental sperm, then you will produce a triploid offspring, and that's bad. Okay? So here you can see that the benefits of this apomictic egg is when mating, during hybrid-hybrid matings, is to produce a nicely diploid egg, right? And that's why also you need the hybrid-hybrid matings, because when you don't have them, then it's bad to do apomixis. So now we are at a new situation where, okay, so you had your Y-type hybrids equilibrium, and a mutant appeared, and it's a great mutant. It invades, it reaches a new equilibrium, and the number of Y-types have reduced a little bit due to competition for resources. And now I'm going to do some fitness maximization, because I want to know where selection and evolution is going to go on the long term, right? And why am I doing fitness maximization? So remember, now we are at a point where the parentals are still at their equilibrium. Now I have an equilibrium Y-type mutant, H2. And I want to put a new mutant, H3, with a new phenotype. And again, I'm going to ask, is this new mutant going to invade? So I have this is really the growth rate of this mutant population. And I can prove that if this is positive, really, what I'm saying is that the growth rate of the new mutant is better than the growth rate of the older mutant. So whoever has the greater growth rate is the mutant that's going to spread and invade. So, and really growth rate, you can see that as fitness. So basically what I'm saying is that a mutant is going to invade if it has a better fitness. Selection is going to maximize fitness, big surprise, right? So that's why I'm going to do maximization of fitness, right? Because that's where selection is going to tend. So to make a maximization, I do partial derivatives around the three phenotypic axes, alpha, beta, and sigma, right? So I have these equations, and actually it's much easier than it looks, because the first equation, it's always greater than 0, which means that f always increases as alpha increases. So f is maximized at the maximum of alpha, which is alpha equal 1, right? So the best thing for selection is alpha equal 1. And the second equation is always greater than 0 when alpha is greater than 1/3, which I know is going to eventually happen because I just proved that fitness is maximized at alpha equal 1. So this is also going to always be greater than 0, and fitness is maximized at beta equal 1, the maximum of beta. And again, the same thing happened with sigma, so what I just proved with that fitness is maximized at the three phenotypic traits at their maximum 1, which is full gynogenesis and complete asexuality, right? So what these equations tell me is that once I have a first mutant that managed to invade, then any new mutant that is even more asexual is going to invade and replace it. And I'm going to have a dynamic like that where, so remember I was here, and now a new mutant invades and replaces the older mutant, and then another one more asexual, and then another one more asexual, until full asexuality has evolved by repeated mutations. Each time the Y-type hybrids decrease in number because of competition for resources, but you can see that they never go extinct because they are always maintained by direct hybridization. But obviously, after some evolutionary time, the parental species probably they're going to stop hybridizing. And at this point, the Y-type hybrid, which is sexual, will completely disappear from the ecosystem, and you will be left with only a fully asexual hybrid, right? Which is the situation you can see nowadays in the Amazon molly and other asexual vertebrates. Right? So I also did this fitness maximization a bit more in terms of numerical simulations. So what I do is I'm at a certain phenotype, right? And I derivate the fitness along the three phenotypic axes. and I take the greater partial derivative, which tells me what is the greater selection coefficient. And for example, if the greater selection coefficient is in direction of alpha, then I'm going to assume that at the next evolutionary step, alpha has increased a little bit, because a mutant that increases alpha has spread, right? And I do this many times, and thus I can kind of recreate what would be the most probable evolutionary route toward asexuality. So I'm going to have something like that, where you can see, well, alpha increases, and then sigma increases, then beta increases a little bit, and sigma a little bit more. This is just an example because I wanted to put something that is visually easy to understand. But actually what you see most of the time for most of the parameter values is something very much more radical, where you have, first of all, alpha that goes all the way to 1. So you have the evolution of apomixis, complete apomixis. Then you have a sigma that goes all the way to 1, fully female biased sex ratios evolve. And finally, you have the full evolution of paternal genome elimination. And I'm going to talk a little bit more about that in the conclusion, but we have a completely stepwise process for the evolution of asexuality in this model. So I'd like just to finish with just the core conclusions. I'm not going to enter into too much details. First of all, I think the most interesting question of that is, what is the selection force that is driving the evolution of asexuality in our model? I want to convince you that it is something that has to do with reproductive assurance. Okay? So I've made a very important assumption on the hybrid sperm. Remember that it's weird and cannot decondense normally in the egg. What that means is that a normal meiotic egg, when it receives a hybrid sperm, it will be like a sterile mating. Nothing good will come from that. But if the egg is apomictic, is diploid, then you produce a viable offspring. And remember, we are in a situation where hybrid mating happen often, relatively often. So this situation of these matings are sterile is going to happen often. But all of a sudden, you have a mutation that brings in apomixis, and you rescue these matings, and you get a better reproductive output. You produce more offspring because you have rescued all these matings that were sterile. Right? And that's why I tend to call this process reproductive assurance, because you gained fertilization opportunities. You gain reproductive output, really. And then, so that's for the first step, right, the evolution of apomixis. And then you have paternal genome elimination. So, okay, apomixis has fully evolved. Now you are in this case. And, okay, that's good. When you mate with hybrids, everything is fine. But still, sometimes, probably, you're going to mate with parental males, right? And in that case, you know that you produce a triploid, and that's probably bad. Really, in nature, it's not always so bad, but it may have a fitness cost. And that's why paternal genome elimination is going to evolve in the second step. Because when you start to do paternal genome elimination, then you rescue these matings that were now bad, right? So that's the same process of reproductive assurance, of gaining fertilization opportunity or mating opportunities, to gaining reproductive output by evolving these traits, right? So I told you in the introduction, we made different models. We made 3, actually. So the model I presented you today is this one. Here in this model, Y-type sexual hybrids could produce viable offspring only when it was mating with parental males, right? But now, once you have the full evolution of asexuality, you can mate with any kind of male, parentals or hybrid males. So you can see that overall, you've gained fertilization opportunities. You will produce more offspring because you can mate with more males, right? Another model, we made another model where the gain in fertilization opportunities is even greater. And in that case, you can see that gynogenesis evolves even more easily and in a similar stepwise process, right? One phenotypic trait at a time. And finally, we made a more normal assumption, let's say, that the sperm of the male, of the hybrid males, is completely non-functional. It's not even produced, right? The males are completely sterile. And if you do this assumption, if you're talking about gynogenesis, what's going to imply that you have no gain in fertilization opportunities when evolving gynogenesis. Y-type hybrids will only produce viable offspring when mating with parental males, and the same happens with the gynogenetic females because they still need a sperm, right? So here you have no gain in fertilization opportunity, you have no reproductive assurance process. Still, you can find a parameter space where asexuality is going to evolve because you have other benefits, classical benefits of asexuality, so it may evolve. But the parameter space where it works is much, much, much reduced, and the three phenotypic traits must evolve at the same time, all at once. So you need a mutation that modifies the three phenotypic traits at the same time, which obviously is much harder, right? So from constructing this model, we can kind of predict that this reproductive assurance process could make a big difference in terms of selection, right? It would really help asexuality to get established. I think the fact that it is stepwise, apomixis first and then paternal genome elimination, is very important because it makes a much easier evolutionary route, evolutionary path. And it happens, again, because we made this core assumption on the hybrid sperm that it does not decondense, and thus an apomictic egg does not necessarily need paternal genome elimination to produce a viable offspring. It can produce a viable offspring without it, just by mating with a hybrid, right? That's really the core assumption of the model. And that allows us to propose for the first time an evolutionary route where phenotypic traits can evolve independently. You don't need to evolve paternal genome elimination and apomixis, let's say, at the same time, which helps any evolutionary process. And the last thing important is obviously this core assumption that I made on the hybrid sperm. First, it's what gives you the benefits of asexuality, because it's what gives you an amplification of fertilization opportunities. And also, it's what is responsible for a stepwise evolution, so one trait at a time, which makes for a much easier evolutionary path. So this core assumption of the hybrid sperm that is weird, of the sperm that is weird, is what makes the whole deal. And obviously, this assumption, you wouldn't expect something like that in a non-hybrid species. Right? You don't expect that all males have weird sperm. If you're talking about a normal species, that wouldn't make any sense. But in a hybrid where we know that you can have genomic incompatibilities, when we have cases of dysfunctional sperm or weirdly functional sperm, it is completely entirely possible. And the other assumption we made in the other model is also possible, right? So really, this selective advantage of reproductive assurance that I tried to convince you that it should make a great deal of a difference should be restricted to hybrids. And that kind of conclude my talk by that, right? And then we can explain at least partly the link between asexuality and, hybridization and asexuality. What I'm going to try to illustrate is that hybrid species are some sort of a fertile ground for the evolution of asexuality. It is a specific context where you have specific selective forces that may favor asexual species. And in normal species, that may not happen, and that may be why you have so many asexual species that are of hybrid origin. And so with that, I'm finished. So I'm just left with thanking my collaborators, so my boss Francisco in Royal Holloway, Jeff in Western Ontario, who helped us a lot with the math, and Waldir and Go, who were in charge of the more empirical part of the project. And so just mentioning that this work was co-funded by NSF in the US and NERC in the UK. So, well, thank you very much for your attention, and I'm ready to take any question you may have. Thanks.
Thank you very much, Frédéric. Questions? Okay, I'll start then. Okay, so in the study species that you were working on, the Amazon molly, that's been around for a long time, so that's at a late stage in the process. You've already got the elimination of the paternal sperm, you've got the sex ratio fixed. Presumably, because different hybrid zones are arising at different points in time, and also the different parental species are coming into contact with each other at different times in different parts of species hybrid zones, there will be systems at different stages in the sort of evolution of asexuality process that you've described. So are there known examples of different stages in that process which haven't yet sort of fixed the asexuality or haven't arrived at the parental sperm elimination and that kind of thing?
So I'm not too aware of that. Obviously, you would need to take the the species right on the right time, right? Because once it's asexual is fixed, then it's fixed, let's say, forever, right? And we're talking like less than 1% of eukaryotes are asexual. So even though it has appeared repeated many times, it's still not so easy or to find like in it. Here I have an asexual, right? But at least in the lab, we know that weird things happen. So at least when you take the F1, you can end up having all sorts of things. So for example, in the Amazon, when they try to recreate the Amazon molly, they would observe, for example, that some capacity to produce diploid eggs, not all eggs, but some, but no paternal genome elimination. And obviously, the route that I showed you, we were starting from pure sexuality at first, right? That's kind of to make the broader assumption possible. But you can take the same process and start from any point between sexuality and asexuality. You can assume that the F1 is already a bit asexual, and you're going to then go through a first phase that you could see like an optimization of asexuality. Like asexuality is a bit produced by genomic incompatibilities, but it's not great, it's not perfect. So you go through a phase in evolution where you're going to perfect it, optimize it, until reaching completely asexual species. I don't have example in mind where you find a species in the wild that is not an F1 and that is not completely asexual, but I would not be surprised that it exists.
Okay, Aida, and then Rafa.
Great talk, thank you. I'm wondering what's the advantage to the males? Why is it maintained? Like if, like reproduction, fertilization, and all that is costly, but their genome is not incorporated into the next generation. So why is that still happening? Why it's not evolving into just a purely female reproducing population?
Okay, yeah. So you mean in the normal gynogenetic Amazon molly, why the males keep mating with this female? So that's also something very interesting when I started to work on this species, because gynogenesis is really weird. In the both sense, like first, why the females do gynogenesis and not just parthenogenesis, and you just don't have to mate with any males anymore, and so you get rid of the cost of mating. It's like gynogenesis is the worst of two worlds, right? You have, let's say, the poor adaptability of asexuality, but you still have the mating costs of sex, so that doesn't make sense, right? We don't know why gynogenesis, really, and not parthenogenesis. We know that there is a clear phylogenetic distribution, so all that I know of, all asexual reptiles that are asexual, all reptiles that are asexuals are parthenogenetic, But for example, parthenogenesis is not present in fishes, and I think in amphibians, I'm not sure. But in fishes, you either have gynogenesis or hybridogenesis. And so these probably point towards some sort of physiological constraint that we don't know. For example, what came to my mind is the idea that, for example, the parthenogenetic lizards, the females, the parthenogenetic lizards, they seem to have some sort of physiological costs. So they manage to survive, but they don't give birth to as many offspring as their sexual counterparts do, right? Because the ancestral species, again, were adapted to sex for a very long time. So when you do asex, all of a sudden, maybe you don't reproduce as well because you don't have. So for example, sometimes there are physiological gifts associated with sperm, so you don't receive that kind of gifts. Anyway, you have different things going on there. But still, there is no definite answer. And then there is the point of view of the males. They are kind of robbed of mating opportunities. The males have no interest in mating with asexual females. As you say, their genome is not transmitted. So why do they keep doing that? And we don't know either. There have been theories and there have been experiments and such, but we don't know. So for example, you could think of, I don't know if that hypothesis was completely discarded or not, but you can think that, so all these species of fish, they live in groups, and you have certainly things like social status. So if you have a male that mates with a lot of females, then the other females may want to mate with that male, guessing that it has a good fitness advantage. It's like, if the other female thought it was a good mate, I'm going to mate with it also, because just in case it's right? So you can have things like that, and we know you have mate copying mechanisms in this species. But yeah, again, we don't have definite answers, and that makes the whole thing so interesting and so weird about gynogenesis. Even in terms of population dynamics, when you do a model with these populations, most of the time it collapses. The population go extinct because, for example, the Amazon molly, they parasitize the parental species for matings, but they also compete for resources. And in terms of population dynamics, that's terrible. So we don't either understand how they keep coexisting. And so these are open questions, really, I guess.
Hi, thank you for the talk. It's a bit related, and sorry if it's a silly question. I'm a microbiologist, so I don't know much about sexual. But I think there are some animals that, although they are not hybrids, they have sexual and asexual reproduction, right? In that case, because your model proposed that asexuality is related to hybridization, have you thought what will be the case for those animals, why asexuality has appeared?
So you mean like cyclical asexuality, right? So I've not worked on that really. We know, I think, if I recall correctly, we know that there are some advantages of cyclical asexuality, because then it's kind of the contrary of gynogenesis, in the sense that you kind of get the best of the two worlds, because you are able at certain moments. So the classical case is that when environment is stable, you produce asexual very, very fast, because it's a process that is really cheap. And then all of a sudden the environment has some big changes, and then you do sex, because then you recreate a big chunk of genetic diversity, and that allows you to adapt to the changes. Or just do sex a little bit from time to time, allows you to maintain a certain level of genetic diversity, which is enough to keep going on. And yet you get the benefits of, most of the time, producing a lot of individual cheap, right? And then there is something, I guess, more interesting there, even more, That is, from these cyclical asexuals, sometimes you have the evolution, the transition towards obligate asexuality. And then that's probably something I'm not sure we understand completely, because if cyclical asexuality is the best of the two worlds, then why do you switch back to complete asexuality? What's the point there, right? And I have no idea, really.
And I have a second question related to your mollies. You mentioned at the beginning that sexuality has some cost, and one is mating. But in your mollies, mating is maintained, and I guess that's costly. And at the end, if the males don't reproduce their genes, they will have some degeneration. They are not selected by the best genes, or short to say, I don't know. Do you consider that mating will disappear eventually, or?
So here in this model, you always have matings, because I wanted to study or take the example of gynogenesis. And gynogenesis, it works like that. You have the matings. I have some hybrid males in my populations. They are produced by either hybridization or backcrossing, or even asexuality, whilst completely female by sex ratio have not evolved, can produce some males. When you don't have that anymore, when you, let's say, full asexuality has evolved in your hybrid population, then you don't produce hybrid males from hybrid mothers, right? They are just produced by direct hybridization, which can produce all kinds of things. And when hybridization stops, then these males will completely go extinct, because they are not produced by anybody, basically. And so then the females, the only choice they have is to mate with the parental species. And which is why also parthenol genome elimination is favored at the very end, because you have evolved apomixis, you have evolved female biased sex ratios, and maybe then you could have the stop of hybridization. So really you need to make the best of mating with parental males. And in that case, to do that, you need parthenol genome elimination, or you will do triploids. But yeah, in the end, when you have a natural system like Amazon molly has no males. It's a purely unisexual fish. Yeah.
Thank you.
And I suppose that means it's this purely gynogenetic species can't exist unless at least one of the two parental species is there. So in an evolutionary sense,
yeah, so
it's a bit of a dead end.
Yeah, that's so. Yeah, so we know that they can get some sperm from other species, but it does not seem to work as well. Really the best is the parental species, because they are so related. That's one of my ideas, one of my projects in the future with that line of investigation, is to understand why gynogenesis and not parthenogenesis, and why one should win over the other. Because at first, when you think about it, gynogenesis seems so maladaptive, right? We've talked about it. So there must be probably some physiological constraint, because the phylogeny is so clear that they are just species that cannot do gynogenesis, that will ever do parthenogenesis, and some others that cannot do parthenogenesis and will never do it. But then, still, so there can be that, and there can be also the case that maybe in some cases, in some life history contexts, in some kinds of environments or reproduction, gynogenesis maybe is better than parthenogenesis. And my idea would be that it may have to do with the adaptation to sex that all these newly asexual species still bear. And it's true that if mating, it's costly, right? But maybe sometimes it's not. Maybe sometimes the cost of matings are not great, because you have mates everywhere and it costs nothing, and you don't have, and it's very fast and whatever. And also sometimes we know that you have, like I said before, for example, gifts associated with the act of mating, which may give a benefit. And the best example of that is the fact that parthenogenetic lizards have a lesser reproductive output than their sexual offspring. So it seems that there is some kind of physiological cost. So sometimes the cost is so big that the species can never evolve asexuality. It makes no sense. but maybe the cost sometimes is little, so parthenogenesis is fine, but gynogenesis is better. And so I'd like to, in the near future, to produce some models and prediction about what would be the contexts where gynogenesis would be better. And can we see that the phylogenetic distribution we have correspond to that context, and does that fit, right? Those are avenues of research that are still open. Yep.
Okay, so if there are no more questions, thank you very much to Frédéric for a very interesting seminar. We can continue the discussion down at the kiosco with a drink. And next week we have another seminar on hybrid zones for all the hybrid zone fans out there. Okay, thank you very much.